Okay, so moving on to the other types of the type 2 and type 3, again, we're still talking about single pass transmembrane proteins. They are shown here. And as we'll see, they use the topogenic sequences that we indicated at the very beginning. The only difference is which topogenic sequence is involved. So for the type 2, keep in mind what has to be accomplished, and that is a C-terminal extracellular domain or luminal domain and an N-terminal that is not processed facing the cytoplasm. So this is essentially an inversion of the orientation from the type 1. And the way that happens is, first of all, by not using a signal sequence at all, and that's why this is not a processed end, but instead what you have is a nascent chain that's somewhere along that sequence, and it can be anywhere. It doesn't make it any difference at all where the topogenic sequence is. But what you're going to have is now a signal anchor sequence somewhere in that nascent chain in order to make a type 2 single pass protein. Single pass means that there's only one signal anchor present in the case of type 2s. So again, what that is a positively charged region, one or more positive amino acid residues followed by a hydrophobic stretch that is about the length of the cytoplasmic membrane, or the ER membrane to start out with. Okay, so that's going to be here. And if you make that at the membrane in the translocon, then there's going to be an intermediate here. So again, the ribosome is going to be right here somewhere. And here is that topogenic sequence. Positive charge at the N-terminal region of that sequence. Negative charges right here that form that salt bridge, and that's why this loop forms. Same as with the signal sequence. The difference is there's no site recognized by signal peptidase. So if you carry this mechanism further, open up the seam, let this region sample the lipid, decide if it likes it. If it does like it, it will move out into the membrane. You no longer need the translocon at all. The protein is now going to be released from the ribosome in this fashion because the initial topogenic sequence dictated this loop. The N-terminus is going to be basically on the side where the ribosome was, and this region is now luminal. And if you follow this mechanism and believe it, then you can see that this region can be as long as you want it, and this region can be as long as you want it. So you can use a type 2 mechanism to make integral membrane proteins single pass again that have very long cytosolic domains, very long extracellular or luminal domains, or both. No restrictions whatsoever. On the other hand, in the case of the so-called type 3, remember what they are. They are integral membrane proteins that have a unprocessed N-terminus, so NH3+, not N', and that N-terminal region is luminal, and the C-terminal region is cytosolic. Now, I really do not like the way the textbook draws that mechanism because this is not what happens. This is what happens, and it's an important point, and that is that here is a stop transfer sequence. Nothing new about it. It's exactly the same one we used in the previous diagram. It's just that in the previous diagram we had a signal sequence that preceded the stop transfer, but now we don't. So we've got the N-terminal nascent chain followed by a stop transfer sequence. So when this thing encounters the translocon, it will stuff that relatively short region of the nascent chain into the pore until the stop transfer sequence gets there, and then you get the standard issue salt bridge between that positive region and the negatively charged ring around the translocon. So it just sticks, and that's where we are right here. This never exists. It's not at all correct. So what happens now is the ribosome is here. This is cytosolic, and again, this can undergo that seam opening and closing. This is hydrophobic, positive followed by hydrophobic. So that migrates into the phospholipid bilayer of the ER, and the ribosome is out here somewhere finishing up the translation. So this can be as long as you want. It can be 1,000 amino acids long, but this has to be short because it has to fit into the translocon. So for example, you couldn't make a domain, a folded region here, and then stuff that through. There's no unfolding mechanism involved in this, so this region has to be short. So for type 3, single pass integral membrane proteins, the N-terminus is unprocessed and short by necessity. And once again, once that is made, you know that this was the original topogenic sequence, which in our case for type 3 is a stop transfer sequence, and it will be positive charged on the cytosolic side and negatively charged on the luminal side. So while we're on that topic, go back to the type 2. We know that this began its life over here as a signal anchor topogenic sequence, but once again, although the orientations of the N and C terminus are flipped relative to type 1 and type 3, positive charge is on the cytosolic side, and negative charge is on the luminal side. And there is a concept or a phrase associated with this that's worth memorizing. I may never say it again, but there's the so-called positive N rule. And you don't have to know what that means. All you got to do is memorize it, because if you memorize it, then you'll get these things straight. Because if you've done if you've made a transmembrane protein and the positive charge is on the outside, then you've done it wrong, because that's not the way it works. So the reason that this rule exists, that is, that biology is not based on a positive out rule, is that if you go way back in your self-phys learning and think in terms of what I wanted you to have running in the back of your mind continuously for the rest of the semester when I brought it up, and that is the sodium-potassium ATPase. So if it's somewhere on this cell, then what it's going to do is end up creating a charge imbalance that puts positive charges here. So what those positive charges do is stabilize this particular arrangement. So they make it a more stable environment to have the so-called positive N rule. And this, again, is applying to the protein, not to the gradient, which is positive out. The positive charges are on the outside. You don't need to memorize that, but you do probably, you would be better off if you memorize positive N as a phrase with respect to these transmembrane proteins. Okay, so we got one more type of integral membrane protein to deal with. That's the so-called type 4. And type 4 is a category that just includes integral membrane proteins that have more than one transmembrane domain. So there is no distinction between N and C terminus location in the case of type 4s. It's just they have more than one transmembrane domain regardless of the overall mechanism. They're all called type 4. And another term for that is much more common than type 4 is polytopic. Now, these are described actually fairly nicely in the Wikipedia description of topology of transmembrane helices. And in that page, they talk about single-pass proteins as being bitopic, B-I-T-O-P-I-C. I don't think I've ever heard that term used by anybody else. But polytopic is fairly common, and it just indicates that there's more than one transmembrane domain in the protein in question. So over on the right-hand side of this diagram are two mechanisms for how that could be achieved using the topogenic sequences that we talked about, the stop transfer and signal anchor sequences. But in fact, you can do a polytopic protein using signal sequences as well, and that's very common. So in that case, what you would have would be, let me get the orientation straight, it's kind of upside down in my way of thinking. But anyway, what you'd have is the lumen and cytosol arranged like here. So beginning in the lumen, you know if there's a signal sequence involved, then the first thing that you would write is N prime, because that's the only way that that's going to happen with the signal sequence. But then what you would have is at least two, but any number of transmembrane regions, make those red just for kicks, to make one point, and that is that these were once topogenic sequences. So the only issue is which topogenic sequences would produce that. So what's missing from this diagram, of course, is the thing that kicked it all off. That's the signal sequence. So that would be the first thing that was encountered during co-translational translocation. If you're going to start with the signal sequence, then this sequence right here has got to be a stop transfer. There's no way you can follow a signal sequence with a signal anchor, and try it and you'll see. And if you realize that that's impossible, then you've understood. You've got it all down. But if you want, you can just trust me that has to be the case. So once you've established that, so this was a signal sequence, this is a stop transfer, then you can make any type of polytopic protein you want. It's just going to have to be the case. The rules are that if this is a stop transfer, this has to be a signal anchor, in which case this will be a stop transfer. And if you want to keep going, so this has 3 transmembrane helices with a processed N-terminus in the lumen and a C-terminus on the cytoplasm. If you want to keep going and bring this back through to the lumen, then what you've got to have is, count them up, stop transfer, signal anchor, stop transfer, signal anchor. And you can do this 10 times, 100 times, although I don't know of any protein that has 100 transmembrane helices, but for sure some have 10. And all of those transmembrane regions were once topogenic sequences, and they all are alternating versions of the two topogenic sequences that we talked about, maybe beginning with the signal sequence. So right here, 1, 2, 3, 4, 5, 6, 7 transmembrane helices with the N-terminus in the lumen, C-terminus as a fairly long cytoplasmic tail. That is an icon, basically, in terms of polytopic proteins. This will be a G protein coupled receptor of the signaling lectures that are going to come up right after vesicular trafficking. So you'll see this icon a lot. But as far as we're concerned today, this protein was formed as a result of co-translational translocation using those alternating topogenic sequences. And once again, your book does weird things. I fix it here, but not here. So this is a stop transfer sequence followed by a signal anchor, followed by a stop, followed by a signal anchor, stop, etc., etc. So that makes the 7 transmembrane helices. This should be longer if that's longer. But in any case, that's how you get a G protein coupled receptor. And then finally, there's another mechanism that produces a double pass that has an orientation that neither of these have, and that is that the N and C terminus are on the cytoplasmic side. So what that tells you is the only way to get that arrangement, an unprocessed N-terminus on the cytoplasmic side, is if you begin with the signal anchor and follow that up by a stop transfer sequence. That's really the only way that could possibly work. So you can work those things out logically if you want to. You can memorize them if you want to. To me, it's easier to just figure it out, because once you figure it out, it's pretty easy.